Imagine a world without genetic diseases. Imagine crops engineered to withstand climate change. This is the promise of genome editing, a revolutionary technology poised to transform life as we know it. At the heart of this revolution lies CRISPR, a tool that allows scientists to precisely edit the very building blocks of life, our genes. Just as a writer edits a text, scientists can now target specific genes, correcting errors or inserting new information. This ability to rewrite the code of life has profound implications for medicine, agriculture, and even the future of our species. But with great power comes great responsibility. As we venture into this uncharted territory, we must carefully consider the ethical dilemmas that genome editing presents. To grasp the significance of genome editing, we must first understand the language of life, DNA. DNA is the blueprint of life, a complex molecule that carries the instructions for building and maintaining an organism. It's made up of 4 chemical bases, represented by the letters A, T, C, and G. These bases pair up in specific ways, forming the iconic double helix structure of DNA. Genes, the functional units of DNA, are like individual sentences within this vast genetic text. Each gene carries the instructions for making a specific protein, which carries out a particular function in the body. Changes in the DNA sequence, known as mutations, can alter the instructions encoded by genes, sometimes leading to disease. CRISPR, short for clustered regularly interspaced short palindromic repeats, is a revolutionary gene editing tool that has taken the scientific world by storm. This groundbreaking technology has opened up new possibilities in genetic research and medicine. Derived from a bacterial defense system, CRISPR allows scientists to make precise cuts in DNA at specific locations. This precision is crucial for the accuracy and effectiveness of genetic modifications. It's like having a pair of molecular scissors that can be programmed to target and edit any gene of interest. This programmability makes it a versatile tool in the hands of researchers. The CRISPR system consists of 2 main components: the Cas9 enzyme, which acts as the molecular scissors, and a guide RNA molecule, which directs the scissors to the target DNA sequence. The guide RNA is designed to be complementary to the specific DNA sequence that needs to be edited. Scientists can design the guide RNA to match any desired DNA sequence, allowing for highly targeted gene editing. This customization is what makes CRISPR so powerful and adaptable. This remarkable precision makes CRISPR a powerful tool for correcting genetic defects, introducing beneficial mutations, and potentially eradicating genetic diseases, and even developing new disease therapies. The potential applications of CRISPR are vast, ranging from agriculture to medicine, and it continues to be a focal point of scientific research and innovation. One of the most promising applications of CRISPR technology is in the field of medicine. Scientists are exploring its potential to treat a wide range of genetic diseases from cystic fibrosis to sickle cell anemia. By correcting the faulty genes that cause these diseases, CRISPR offers hope for cures where previously there were none. CRISPR's potential extends beyond inherited diseases. Scientists are investigating its use in fighting cancer by engineering immune cells to target and destroy tumor cells more effectively. Clinical trials are underway to evaluate CRISPR-based therapies for various diseases, and early results have been promising. While challenges remain, CRISPR holds immense promise for revolutionizing the way we treat and potentially cure some of humanity's most devastating diseases. Section 5. CRISPR in the field, feeding the world. Beyond medicine, CRISPR technology has the potential to transform agriculture and address global food security challenges. This revolutionary tool allows for precise modifications in the DNA of plants, opening up new possibilities for crop improvement. By editing the genes of crops, scientists can enhance desirable traits such as yield, nutritional content, and resistance to pests and diseases. This means that farmers can grow more food with fewer resources, making agriculture more efficient. Imagine crops that can thrive in drought conditions, resist devastating plant viruses, or provide increased nutritional value to combat malnutrition. These advancements could be game changers for regions struggling with food scarcity. CRISPR-edited crops are already being developed and tested, offering a glimpse into a future where agriculture is more sustainable and resilient. These innovations promise to reduce the environmental footprint of farming. By increasing crop yields and reducing reliance on pesticides and herbicides, CRISPR could help feed a growing global population while minimizing environmental impact. This could lead to healthier ecosystems and more sustainable farming practices. However, the use of CRISPR in agriculture also raises concerns about biodiversity, ethical considerations regarding genetically modified organisms, and the potential for unintended ecological consequences. It is crucial to address these issues through careful regulation and ongoing research. Section 6, the designer baby dilemma, promise or peril. Perhaps the most ethically charged application of CRISPR is the potential to edit the human germline, the DNA that is passed down from one generation to the next. While this could potentially eliminate inherited diseases, it also raises the specter of designer babies, where parents could choose their children's traits, such as eye color, height, or even intelligence. The possibility of germline editing raises profound ethical questions about human autonomy, reproductive rights, and the very definition of what it means to be human. Critics argue that it could exacerbate social inequalities, create a genetic divide between the enhanced and the unenhanced, and have unforeseen consequences for future generations. Section 7. Unforeseen consequences. Tampering with nature's blueprint. One of the most significant concerns surrounding genome editing is the potential for unintended consequences. The complexity of biological systems means that even small changes to DNA can have ripple effects throughout the genome, leading to unforeseen and potentially harmful outcomes. Off-target effects, where CRISPR edits DNA at unintended locations, could have unpredictable and potentially dangerous consequences. Moreover, the long-term effects of germline editing on future generations are unknown and raise ethical concerns about experimenting with the very essence of human life. While CRISPR is a powerful tool for good, it's crucial to proceed with caution, acknowledging the potential for unintended consequences and the need for rigorous safety and ethical oversight. Section 8. Navigating the ethical labyrinth. Regulations and guidelines. The ethical dilemmas posed by genome editing have sparked intense debate worldwide. Many countries have strict regulations or outright bans on germline editing, recognizing the profound ethical and societal implications of altering the human germline. Scientific organizations and bioethicists have called for a moratorium on germline editing until the ethical and societal implications are thoroughly considered. International collaboration and open dialogue are crucial for establishing ethical guidelines and regulations that govern the responsible use of this powerful technology. The future of genome editing hinges on our ability to balance scientific progress with ethical considerations, ensuring that this revolutionary technology benefits humanity without compromising our values or future generations. Section 9. The future of genome editing. A world transformed. Genome editing, particularly with CRISPR technology, holds immense promise for revolutionizing medicine, agriculture, and our understanding of life itself. This groundbreaking technology allows scientists to make precise changes to the DNA of living organisms, opening up possibilities we could only dream of a few decades ago. The potential benefits are vast, from curing genetic diseases to feeding a growing population. Imagine a world where inherited conditions like cystic fibrosis or sickle cell anemia could be eradicated, or where crops are engineered to withstand harsh climates and pests, ensuring food security for all. However, the ethical challenges are equally significant, demanding careful consideration and responsible stewardship of this powerful technology. Questions about consent, accessibility, and long-term impacts on ecosystems and human health must be addressed. As we venture further into the uncharted territory of genome editing, we must proceed with caution and humility. The potential for unintended consequences is real, and the stakes are incredibly high. Open dialogue, ethical guidelines, and robust regulations are essential to ensure that this transformative technology is used responsibly for the benefit of all humankind. Collaboration between scientists, policymakers, and the public is crucial. The future of genome editing is in our hands. It is up to us to navigate this complex landscape wisely. By embracing both the promise and the peril, we can harness its power to create a healthier, more sustainable, and more equitable future for generations to come. The journey ahead is challenging, but the rewards could be extraordinary.
So hit that subscribe button, ring the bell for notifications, and let's turn the page on a new adventure together. Get ready to experience books like never before, only on VBook 360. Like, comment, and share to support the channel and keep the video magic alive. Thank you for stopping by. And remember, every video has a story, and every story deserves to be shared. See you in the next video.